Hi everyone, in today's video I'm going to be going over Lab 8 growth kinetics via the MTS assay. So as always, I'm asking you guys to go ahead and read through the pre-lab reading and kind of get an idea for the basic theory behind the lab. One thing I just want to remind you guys is that this is a population assay, which means you're measuring individual cells, but you're measuring an entire population and then making adjustments and calculations for an average. Okay, and this is going to be important because next week we're going to move on to some individual cell assays, and that will be quite interesting. So I'm going to try to make this video a little faster so at the end I can go over some tips because I've realized that there have been some problems with finishing the labs on time. So without further ado, let's get started. One thing I want to really stress, and this is one thing I've kind of observed, is that I think you guys can be splitting into partners more often and doing things in parallel. So someone can be trypsinizing cells off the 96 well plate, and someone can be trypsinizing the T25, or counting while someone's passaging or culturing. And you guys should really do this because otherwise the labs are going to take way too long, and that is not good. So don't be afraid to separate. You guys are, I don't know, well prepared for this. So yeah, do that. Make sure you guys get through everything on time. Specifically, right, just to clarify these things and kind of draw it out, you've seeded a 96 well plate with 4 wells for 2 different conditions. What you're going to be doing in this lab is taking one of those wells, counting it, and then the other 3 wells are going to go for your MTS assay itself. Okay, so I'll just kind of draw that in here. In addition, you need to make standards. So with your T25, what you're going to do is take that, passage that, and then seed those guys onto another, I think, 6 wells for standards. Okay, so in total, you'll be doing 9 times 2, 18 wells for MTS. Just kidding, 12 wells of the MTS reagent. Okay? So I believe this procedure is pretty straightforward. One thing I want to point out is that when you're removing them, after you've trypsinized them and kind of, and after you have deactivated the trypsin reagent with your 200 microliters of media in step 3, okay, you can just directly take that 300 microliter solution and put that 10 microliters of that into your hemocytometer. And that should hopefully be dilute enough where you can actually get good counts off. Otherwise, what you'll need to do is spin it down and try to remove as much as you can and resuspend in a smaller volume. But hopefully this 300 microliters is enough. Anyways, let's keep going. So after that, you're going to want to collect your cell titer 96 aqueous 1 reagent. It's going to be frozen, and you're going to want to keep it from as much light as possible. So just make sure that you have aluminum foil or something and you don't have it blasting under light the entire time. This part's pretty straightforward as well. And the biggest thing of this lab that will probably be the most difficult is just practicing pipetting your solutions in. And so right now, while you're at home, you should draw in where your wells are on this 96 well plate. I believe you guys are sharing with a few other groups. So if these are your conditions right here, then you can just draw in your standards like right here. Standard A, B, C, D, E, and F. Okay, so now that we've finished that, I just want to talk about a few things that I've noticed that I think are making the labs go a little longer than necessary. And the main thing was the thing I kind of mentioned before, where even though you guys have 2 people, it seems like that a lot of you guys are or a lot of you guys are just watching one person do stuff. So this guy's doing stuff and you're kind of just watching. Make sure that you're kind of splitting your tasks and time well, and so that when one person is doing one procedure, another person is doing another procedure, and so forth. And the other thing I realized, and this is my mistake, so my apologies, is that the centrifuge that some of you have been using for cells, I told you guys to spin at 1.5 RCF. That's actually wrong. You should be spinning at 2 RCF on that particular machine. It's the smaller benchtop centrifuge, so sorry about that, but make sure you see that change. I've also included tape on the actual centrifuge itself detailing this change. And one third thing that I've kind of noticed is that some people are getting contamination. Just make sure that when you're aliquoting in your pen strep and everything else, that you are doing it at the proper concentration. It should be 100x, which means that you're putting in 500 microliters for your 50 mil conical. I think that's everything for now. If something else comes up, I might make another video or send out an announcement, but hopefully this helps you guys finish up all your labs. So until next time.